Our project looked at lymphocyte detection in ovarian biopsies, which was a multi-member, multi-major AI project from the AI Club here on campus with a wide variety of majors looking at medical images and artificial intelligence. And it aimed to build off some previous research that looked at identifying key regions in human biopsies by training a model to specifically identify lymphocytes, as most of these other cell identification algorithms had trouble classifying this cell.
The current method of detection for ovarian cancer starts with a doctor-ordered biopsy, as seen in the top left. After samples are extracted from the ovaries, they are then preserved in hot paraffin wax and then H&E stained in order to see the cell structure, as seen in the purple-hued samples on the right side of the slide. The samples must then be deparaffinized before being viewed by a pathologist who will annotate those samples for many different cell types. However, the deparaffinization process is quite risky as it has the potential to damage the tissue, and since a pathologist is annotating these samples, we enter the realm of human error. Ideally, another method of detection using artificial intelligence would help to mitigate any human error and get rid of the deparaffinization risks. Previous modern research has presented another method of detection for ovarian cancers. After the biopsy, the samples are preserved similar to before in hot paraffin wax and then put into a Fourier transform infrared spectroscopy machine, or an FTIR machine, which can be seen in the top right. This machine creates a spectrum of absorbances for each sample and then produces a blue hyperspectral image when viewed at a particular band of that spectrum, and those can be seen in the bottom left. This method of detection from previous research allows an AI model to predict on the hyperspectral imaging, producing an AI annotated overlay as seen in the image in the bottom right. But this image in the bottom right is detecting every cell type a pathologist would, while our research is mainly focused on detecting lymphocytes specifically.
So applying that modern solution, we utilized ROSIE to achieve our goal of detecting lymphocytes from that hyperspectral image. So we first annotated our biopsies. We then split the data for training and testing. And finally, we ran our data through multiple classical models, along with our deep learning model, and compared the prediction accuracy. When it comes to our actual data, we use the annotated stained samples as a reference to annotating specific features on the hyperspectral image. As stated before, a pathologist had identified the different types of tissue cores. We then use this as reference to accurately annotate lymphocyte and non-lymphocyte type tissues to ready our data for testing and training purposes.
As we're primarily using supervised learning models for the project, we had to first label our data. On the left side of the screen, there's a zoomed in image of one of the bands of the hyperspectral image with hand-drawn labels overlaid onto it of the lymphocytes. To determine where these lymphocytes are, we cross-referenced with the stained cores that were labeled by pathologists. And on the right-hand side of the screen, you can see some of our current research, which involves using PCA to assist with this labeling process.
Another way ROSIE proved key in our research project was visualizing our data. We needed to make sure that our data was not just biasing to a single biopsy of a patient, but instead generalizing to multiple ones. In order to show this, we graphed all of our data out on this picture you see in front of you. We then ran a k-means clustering algorithm on ROSIE to group together all of the annotations on a single biopsy. This allowed us not only to see the dispersion of our annotations, but it also allowed us to see the magnitude on each biopsy, thus proving that we were not just biasing to a single one, but generalizing to multiple.
Originally, we used a random data splitting function for the test-train split. After initially training on our labeled data, it became clear that there was some overfitting going on with some of the classical models. To better validate our models, we effectively cut our dataset in half spatially. Data on the left half of the set is used for training, while data on the right half is used for testing. This allows us to more effectively evaluate models and is more similar to a clinical scenario.
To more quantify the results of this data split, we're going to look at 3 of the 6 classical models that achieve some noteworthy results, which include the SVM, MLP, and nearest neighbors algorithms. So with the SVM, it actually improved after the data split occurs, which truthfully, we don't have a concrete reason for this, but we believe that because it makes that high dimensional plane to look for the more extreme features in the data set, noisy data was luckily cut out after that data split occurred. And then with the MOP, it performed the best with the highest accuracy in both the pre-data split and post-data split subsets. And then looking at nearest neighbors, suffering 20% accuracy decrease because it references those examples from training to compare against testing data it sees, and because the data split cut out that adjacent pixels being tested, it suffered greatly from this.
After trying out some classical models, we decided to move on to deep learning. As this is an image classification problem, we picked out a CNN architecture. Here in front of us, you can see our first iteration of that CNN. Initially, it got an accuracy of 99.7% before the data split. However, this was very unrealistic. Still though, as we applied the data split, it held on to a 92.5% accuracy, which was our best model yet. Because this model did so good, we are currently in the process of trying out some different layer combinations as well as general hyperparameter tuning in hopes of increasing that accuracy. One thing to note is that during training and inference, we cannot hope to run this on consumer-grade GPUs and hardware as it takes up way too much RAM. Luckily, we have ROSIE.
Let's look at some predictions to see what the 92.5% accuracy actually looks like. On the left, we have an annotated H&E stained image with lymphocyte areas circled in orange. On the right, we have a 500 by 500 pixel prediction from the model, with areas in white representing lymphocyte predictions. This takes about 20 minutes for inference. From this preliminary prediction, the model seems to do an okay job of identifying areas with lymphocytes, but isn't very precise. To fix this, we're looking at doing more annotations and doing more experiments with our models.
As a way to compile what we wouldn't have been able to accomplish without the computing power of ROSIE, or at least in a reasonable amount of time, we pre-processed over 500 gigabytes of cancer patient data, and we were able to pivot incredibly quickly as we were training different classification models, only needing to wait minutes to see how our code negatively or positively affected our accuracy. And from this, we were then also able to predict what our models actually saw quickly too, looking at a sample of our current CNN's results on that bottom right image. Basically, without ROSIE being so accessible, this research project would have likely never come up. This large amount of data is infeasible for even our modern laptops. We were then also able to quickly experiment with our different methods to aid the ongoing process of identifying cell regions in cancer patients faster and more reliably.